Okay, so Krebs cycle. In order to understand how Krebs cycle works, we've got to look a little bit at the mitochondria. So, a quick look in here. If you remember, you've got the outer membrane, as you can see here, the inner membrane. So between that you've got the intermembrane space, which is very, very important in terms of the hydrogen ions. That's why they pump to it to maintain that electrochemical gradient. You've got the matrix, which contains lots of enzymes. You've got the cristae, which provide large surface area inside the mitochondria for the ATP synthase enzymes to exist upon. Now, glycolysis, that we learned about last week, happens anywhere in the cell, well, really in the cytoplasm, whereas Krebs cycle is in the matrix. Again, that's a typical exam question. Where does Krebs cycle occur? In the matrix, whereas glycolysis is in the cytoplasm. What Krebs cycle is, I'm going to have a read. Basically, sometimes called the TCA cycle, citric acid cycle or whatever. It's basically a series of decarboxylations, which you know means removal of carbon dioxide, or a series of dehydrogenations, which is the removal of hydrogen. Ultimately, what's produced? Carbon dioxide, which you breathe out, and reduced NAD, which you've seen from chemiosmosis is very, very important because it provides the hydrogen atoms, which are then split into hydrogen ions in order to drive ATP synthase for chemiosmosis. So it starts with acetyl coenzyme A, which is 2 carbon. That joins to oxaloacetate, which is a 4 carbon compound. 2 plus 4 makes 6, and what you form is 6 carbon citrate. So as you can see from that, acetyl CoA, which is coming from the link reaction, joins to oxaloacetate and then citrate to form the 6 carbon compound. Now, you need to know the names of those 3 compounds. You don't need to know the rest. What you do need to know is the order. So from citrate, the compound then undergoes some changes. It still remains with 6 carbons, so it's a slight structural change. From there, it's decarboxylated to 5 carbons, decarboxylated to 4 carbons, and then the numbers are 4, 4, 4, and back to oxaloacetate. So there's a way to remember it, which I'll show you shortly. And that overall, that is Krebs cycle. The difficult bits are noting where does this occur, the decarboxylation. Well, that's not so bad, because look, this has got 6 carbons. To turn a 6 carbon compound to a 5 carbon compound, you've basically got to take carbon away, so that's decarboxylation. And then again, to turn a 5 carbon compound into a 4 carbon compound, decarboxylation. So that's not too bad. The bit that's difficult to remember is where the hydrogen's taken out. So you've got hydrogen taken out there for NAD, hydrogen taken out there for NAD, and one here for NAD. This one here is FAD, which is flavin adenine dinucleotide. Same as NADH, except it's got flavin instead of nicotinamide. You don't really need to know the details of it. We'll show you where that's relevant later on. In order to remember that, for me, that's a look, cover, check, repeat. Just keep going over it, drawing it out, try and remember it. The other thing to note, look, at this point here, enough energy is produced in that change to produce 1 molecule of ATP. Okay. Again, like I said, I think this is very much a look, cover, check, repeat exercise, just to make sure that you do remember it, and then that will structure exam answers when it comes to writing it in prose. How I like to remember it, well, this is Krebs cabs. The numbers, because you need to know oxaloacetate and citrate, and then the numbers of carbons, and you remember it by 6, 5 and then 4 4s. It sounds like a cab company, doesn't it? So that, for me personally, that's been the most effective way of remembering it. If you want to know all the detail, this is all the detail. Look, that's the citric acid cycle. So what you've got here, citrate and oxaloacetate, you already know about. Acetyl coenzyme A coming from the top, isocitrate, alpha ketoglutamate, succinyl coenzyme A, succinate, fumarate and malate, but you don't need to know them. If you're really into it, by all means learn them, but you're just not going to be asked all of that information in the exam. You're just not going to need to know all of that. Alright. For me, really, if you remember the cab number 6544s, that will help you structure it, hopefully, so you're fine with it. I'm not going to show you this video, it goes into more detail than we need, so I've changed my mind about that. If you do want to see it, it's on the Prezi. I'll embed the Prezi in the Moodle as well, so you can have a look. Okay, that's Krebs cabs. So this is what we're looking at overall then. So oxidative phosphorylation, this section here. Chemiosmosis, you know, we've covered now link reaction, we already knew glycolysis. We've just done the Krebs cycle. Again, I don't expect you to know that off by heart when you come in, but that's one we just need to practice over and over again to get it clear in our head, so that ultimately you can write a 5-6 mark answer for it. We'll go now have a quick look at electron transport chain. Okay, I'm going to skip that video for now because I think this is the easiest way to get your head around electron transport chain. What it is, it's a series of carrier molecules. Sometimes they're called cytochromes, but basically they're passing hydrogen and then electrons down a series of carriers, and in doing that energy is released. If you have a look at this, this is the best way for me to remember it. If you imagine this is an increase in energy level up here, and what you've got is NADH coming in, which is a carrier with hydrogen attached to it. It's quite a high energy molecule with this hydrogen attached to it, and what it's doing is it's basically passing the hydrogen onto another hydrogen carrier. Again, you don't need to know all the names of them, but you do need to know that this second hydrogen carrier is at a lower energy level than the first one. So as that's passed down, I put this star here because enough energy effectively is released to pump 3 hydrogen ions into the intermembrane space. Now if we can get 3 hydrogen ions into the space, what can we make? 1 ATP. So I suppose each of those stars really is representative of enough energy to make 1 ATP. So, as I said earlier, NAD, higher energy level than this one. Energy's released there, so we can pump these 3 hydrogen ions in the membrane space. Again, the hydrogen is then passed down to this next carrier, and then at this point something different happens. The hydrogen is split. So you've got your hydrogen ion here, your proton effectively, because hydrogen, remember, is only made of 1 proton and 1 electron. It's split. So your hydrogen ion goes off, the electron is then passed down here to a lower energy level, and then passed again to a lower energy level, and to another lower energy level. And again, at each of these points here, enough energy is given out to pump 3 hydrogen ions in the intermembrane space, which is what's going on here, and therefore make 1 molecule of ATP. At the end of this, so here what we've got now is an electron carrier with an electron on it. It needs to get rid of that electron. And what does it do? Well, it adds it to oxygen. Oxygen has an affinity for electrons, so it will pick that electron up quite happily and it'll give it a negative charge. Now because it's got a negative charge, ultimately when these hydrogen ions here, they've gone through the intermembrane space, they've gone through ATPs and so on, we've got hydrogen ions with a positive charge. Well, opposites attract, don't they? So the positively charged hydrogen ion and a negatively charged oxygen ion attract, and so ultimately we form H2O, water. You'll sometimes see this, well, shown differently in the books, because that doesn't balance, does it? 1 oxygen, 1 hydrogen doesn't make H2O. It doesn't work like that. So they often talk about it being half H2O and whatnot, but the concept is what you need to grasp. So it's oxygen that you've breathed in, that's the purpose of breathing. The oxygen you've breathed in accepts this electron, then joins to hydrogen, and ultimately that reaction forms water that you then breathe out. So the whole purpose in taking in oxygen is basically to soak up these electrons turn it into water so you can then breathe it out. Okay, again, I'm not expecting you to have all this information stored in your minds ready for coming into the lesson, but a fair understanding of what it is we're looking at will go a long way. I think this is something you need to write out. Once you've got your head around the concept, that's the hard part, but then just remembering it so this can structure your answer is what we're aiming to do. And ultimately, I need you to be able to link it to the whole of the process, so particularly that part there about the hydrogen ions and what is going on there, where they go, which you already know, they head off to chemiosmosis. Let me just pause that there. The thing I'm showing you at the minute is something that really there's always a lot of debate about. When you look in the textbooks, they're all slightly different, but one of the tasks that we'll do when you come into the lesson is working out how much ATP is used here in each of these processes and how much is made, and therefore what the net ATP gain is. You see different figures in different books, so if you do have the time, have a look, see if you can do a bit of research and see if you can figure it out. But we will spend a little bit of time. I'll make an activity that gets you to go through and count those up in the lesson. Okay, so back to the outcomes. These were the aims. So for A and A star, hopefully you can explain how hydrogen ions are moved into intermembrane space. I've not explained exactly, but if you link to some of the things from last lesson, you know where the hydrogen ions now come from in the electron transport chain, and we mentioned the proton pump. You've now seen where the energy comes from to drive that proton pump to get the hydrogen ions into the intermembrane space. Obviously, it's the electron transport chain that provides that energy. Describing the Krebs cycle. So remembering the names of the key parts. So you have acetyl coenzyme A, oxaloacetate, citrate, and then Krebs carbs 6544s. The dehydrogenations and the decarboxylations. And finally, recalling how ATP is made is building up on last lesson really, and looking at chemiosmosis and linking chemiosmosis to the rest of the process. And that's about it for today. There is just one more thing that's just popped into my head. So for each NAD that you've got coming into electron transport chain, how many ATP can you make? So if that's enough energy to put 3 hydrogen ions into the intermembrane space, you know that 3 hydrogen ions equals 1 ATP. So effectively, for each NAD, you get 1, 2, 3. Yeah. FAD that you saw in Krebs cycle, FAD feeds in here. So for each FAD, you only make 2 ATP. Okay. So every NAD, you're basically getting 3, 6, 9 hydrogen ions in the intermembrane space. For FAD, you're getting 6, which is the equivalent to 3 ATP for NAD, 2 ATP for FAD.